In this class, we'll be working with the DNA that you extract from a plant to isolate a specific gene to work with. We need to review the basic DNA structure and function before we start our work. DNA is an organic molecule. This means that it is carbon-based. In addition, organic molecules are associated with living organisms. There are 4 classes of organic molecules. Carbohydrates like glucose or starch, which are primarily used as energy sources or used structurally. Lipids, like this phospholipid, which is used in the formation of cell membranes. Proteins, which are the workhorses of all biological systems. And nucleic acids, of which DNA is an example of. We'll focus our attention on DNA, or deoxyribonucleic acid. This molecule should be familiar to you in structure and in function. The central dogma of molecular genetics focuses on DNA's ability to make copies of itself, and also its ability to be used as a template for RNA synthesis, which in turn is used as a template for the primary structure of proteins. DNA is what's organized and divided during mitosis, which is cell division, or meiosis, which is the production of gametes. Cells pass along their DNA to their daughter cells. Replication of the DNA occurs before cell division. In sexual reproduction, the DNA of 2 separate organisms combine to produce a unique combination of DNA in the offspring. Related offspring, such as these piglets, will have similar and yet different DNA. Transcription and translation convert the information encoded within the DNA molecules into RNA and protein. Therefore, these processes regulate biological function of an organism. Transcription occurs in eukaryotic cells in the nucleus and produces RNA made from the DNA template. This RNA is processed and then in the cytoplasm is used for translation, the production of protein using an RNA template. In most circumstances, the flow of information in living organisms is from DNA to RNA and then to protein. The primary structure of proteins dictates its secondary and tertiary structures and plays a critical part in the function of that protein. DNA is made from nucleotide monomers containing a deoxyribose sugar, a phosphate group, and a nitrogenous base. There are 4 nitrogenous bases, adenine and thymine, which in the DNA molecule will be hydrogen bound to each other, and guanine and cytosine, which also will be bound to each other in the DNA molecule. Chains of nucleotides are assembled through dehydration synthesis. These chains have direction indicated by the 3' to 5' designation. This directionality is critical to DNA's replication and transcription. The backbone of the DNA strand within the double helix is made from alternating sugar and phosphates. The nitrogenous bases extend from this backbone. The hydrogen bonding of 2 strands connects 2 nucleotide strands, which will run anti-parallel to each other. The strands will be complementary to each other, and they will form the well-known double helix. We will consider what a gene is later on when we analyze our DNA sequence from our plants. As you remember from biology, exons are portions of the gene which will be expressed within the protein, and introns are cut out within the spliceosome. This concludes our review of DNA structure and function.